Hello, my name is Leah Dean, and today is April 15th, 2025. Let's go ahead and get the presentation started. Okay, today I will be discussing Chapter 24 of Harper's Illustrated Biochemistry 31st edition, Metabolism of Acylglycerol and Sphingolipids. Lipids are valuable biological molecules with a variety of roles in human physiology. They are not only valuable long-term energy storage molecules, but also very important components of cellular membranes and signaling mechanisms. Some lipids, including acylglycerols and sphingolipids, are particularly important in their biochemistry and physiological importance. This presentation outlines their synthesis, remodeling, and degradation, and subsequently discussing their disorders with clinical relevance. The prominent lipid classes discussed in the presentation are triacylglycerols, tags, phosphoglycerols, and sphingolipids. Tags are predominantly for energy storage. Phosphoglycerols contribute to the membrane structure, and sphingolipids contribute to both membrane structure and cell signaling. Synthesis of acylglycerols begins with glycerol 3-phosphate, G3P, which is produced from glycolysis via dihydroxyacetone phosphate, DHAP, or from the phosphorylation of glycerol directly without glycolysis. This G3P phosphate later gets acylated by GPAT and AGPAT to form phosphatic acid, which is an important intermediary in lipid metabolism. Pathway divergence at phosphatidate can be used to build membrane phospholipids, such as phosphatidylcholine or cardiolipin, or it can be dephosphorylated and turned into DAG by phosphatidate phosphatase, PAP. DAG appears in the pathway and can be acylated by DGAT to form tags for storage in adipose tissue as a source of available in time of energy needs. Tags stored in adipose tissue are not without order, though. Tags are divided between lipid droplets. These organelles, which are presented as lipid storage containers in adipocytes or other tissues, are covered by paralipins, which are protective proteins that mediate the availability of lipase on tags and act as regulators of lipolysis. Some phospholipids, such as plasminogen and platelet activating factor, PAF, are recorded as being products from either link synthesis using DHAP. Plasminogen is abundant in the brain and heart and protects membranes from instability and oxidative damage, while PAF is a strong lipid mediator involved in the inflammatory response as well as other cellular events. For general phospholipid biosynthesis, CDP-activated intermediates are important metabolic precursors that are present in molecules such as phosphatidylcholine, PC, phosphatidylethanolamine, PE, and phosphatidylserine, PS. The body can also utilize alternative salvage metabolic pathways to reach libraries of lipids. For instance, the liver can methylate phosphatidylethanolamine to phosphatidylcholine, which is significant for very low density lipoprotein secretions. Phosphatidylinositol is involved in phosphorylation signaling pathways. However, CDP-activated diacylglycerol is also an important precursor for phosphatidylinositol and cardiolipin, which are important for maintaining mitochondrial membrane integrity. Sphingolipid biosynthesis starts with the condensation of palmitoyl-CoA and serine, mediated by serine-palmitoyl transferase, to form 3-ketosphingolene. The 3-ketosphingolene is reduced and acylated to produce ceramide. Ceramide is very important because it serves as the backbone of all sphingolipids. After forming ceramide, sphingomyelin glycosphingolipids such as cerebrosides or gangliosides can be synthesized, and the latter containing sugar residues. Sphingolipids hold significance, not only for structural matters, especially in nerve cells, but also for cell signaling, with gangliosides particularly carrying functional importance in immune recognition as antigens and receptors. Additionally, the Land cycle is another important pathway for lipid maintenance and adjusts to lipid composition via membrane remodeling. The Land cycle utilizes phospholipases and acyltransferases to remove and replace fatty acids to preserve membrane fluidity and composition, and is heavily active in illness and sickness. The metabolic turnover of phospholipids seem to play a role in the process of signal transduction. For example, the enzyme phospholipase C degraded phosphatidyllinositol into diacylglycerol, and inositol triphosphate IP3 and the messenger feedback signals could promote activation of protein kinase C and release of calcium. With respect to arachidonic acid metabolism, phospholipase A2 could yield arachidonic acid needed to form eicosanoids, like prostaglandins that regulate inflammatory response. Moreover, there is degradation of sphingolipids in phospholipid metabolism that occurred in the lysosome, and this process has sphingomyelinase remove the phosphocholine head group from sphingomyelin to form ceramide. Ceramidase would then hydrolyze the ceramide into sphingosine and fatty acid. In the case of an absence of these enzymes and the performance of the lysosome in lipid metabolism, there would be a circumstance that gave rise to lysosomal storage diseases, such as Tay-Sachs disease and Gaucher disease. This pathology is characterized by lipid accumulation along lipid catabolism pathways of complex lipids that result in the accumulation of lipid in tissue, especially in nervous tissue. An acute condition related to lipid metabolism is infant respiratory distress syndrome, IRDS, due to the lack of lung surfactant. The lung surfactant is primarily dipalmityl phosphatidylcholine. Metabolic disorders are clinically relevant for lipid metabolism disorders. Tay-Sachs disease arises from a deficiency of the enzyme hexosaminidase A, and GM2 ganglioside accumulates in neuronal tissue. Gaucher disease arises from the deficiency of the enzyme glucocerebrosidase, and there was marked macrophages that were filled with lipid vesicles. In addition, regulation by hormones of lipid metabolism especially stimulates extensive numbers of tightly controlled lipid metabolites and metabolism. Insulin phospholipid metabolism in cells signaling favors lipogenesis, fat storage, in times of excess nutrient intake. Glucagon and epinephrine stimulate intra and extramembranous lipolysis to encourage energy demands. Furthermore, slight to moderate elevations in malonyl-CoA seem to prevent the oxidation of fatty acids by CPT1, carnitine palmitoyltransferase 1, when fatty acid metabolism is in a synthetic mode. The intent of this review was to emphasize the importance of lipid metabolism not just as storage, but simply essential towards human health. Lipids serve the body in an array of activities that are more than balancing the resources afforded to the body through storage. For example, lipids provide support for membrane architecture, and lipids even participate in signal transmission. This leaves a significant meaning, and this is why processes produced with dysfunctions in lipid metabolism could lead to significant metabolic or neurodegenerative diseases. Given that, there is an advanced research being engaged to develop therapies. For example, human gene editing, lipid delivery systems to better treat or cure disorders related to lipid metabolism. Now we will answer a few questions. The first question is, what is the primary function of triacylglycerol tags in the body? A, membrane structure, B, cell signaling, C, energy storage, and D, synthesis of sphingolipids. Energy storage would be the correct answer for number 1. Which enzyme catalyzes the conversion of phosphatidate PA to diacylglycerol DAG and triacylglycerol synthesis? Would it be A, glycerol 3-phosphate acyltransferase, B, phosphatidate phosphatase, C, 1-acylglycerol 3-phosphate acyltransferase, D, diacylglycerol acyltransferase? The correct answer would be B, PAP for number 2. Number 3, in the synthesis of sphingolipids, which compound is formed first from palmitoyl-CoA and serine? A, ceramide, B, sphingomyelin, C, 3-keto-sphingonine, D, glycosphingolipids. For number 3, C would be correct, 3-keto-sphingonine. Okay, number 4. What is a common result of defects in enzymes involved in sphingolipid metabolism? A, increased production of phospholipids, B, lysosomal storage diseases like Tay-Sachs and Gaucher, C, enhanced cell membrane stability, or D, decreased fatty acid oxidation? The correct answer would be B, lysosomal storage disease like Tay-Sachs and Gaucher disease. Okay, for the last question, which of the following is a potential future direction in lipid metabolism research? A, development of lipid-based drugs for metabolic diseases. B, increasing the synthesis of sphingolipids. C, enhancing the breakdown of triacylglycerols for energy storage. D, curing cancer. A would be the correct one for number 5. There are the answers, if you'd like to see them all together. Thank you so much for watching, and that is the end of my presentation.